Hey guys, thank you to Telltale for having me today. You're probably wondering, who is this stupid stick figure? Give us Telltale back. Well, there's no need to worry. Telltale's doing quite well over on my channel. Definitely nothing fishy going on over there. So now that I have your attention, we're going to be talking about some very interesting things. Here's a creationist video I found that attempts to debunk evolution with the Komodo dragon. Yep, you heard that right. I was immediately interested, so I had to make a debunking video. Buckle up, guys.
Komodo dragons are the largest lizards. They can reach 10 feet in length and weigh up to 200 pounds.
Ah yes, the Komodo dragon. They are much bigger than your average lizard, and their size allowed them to thrive quite well in their ecosystems and easily compete with other species. They eat quite a few different animals, sometimes including humans. Oh, you think I'm joking?
They have forked tongues like snakes and uses its tongue to detect a prey by smell more than 4 miles away.
Indeed, the Komodo dragon loves to just taste the air, you know. It'll detect you from miles away, you better watch out, it's coming for you.
They can rapidly increase their metabolism to near mammalian levels for amazing bursts of speed and even long strenuous runs.
You better start hitting the gym now, otherwise it'll outrun you.
Komodo dragons have reddish saliva that contains 50 different types of bacteria. Only one bite induces blood poisoning of their prey.
Their bites are dangerous too. How much better could this get? Just a small nitpick, they inject proteins through their bites, not bacteria. Although their teeth are probably inhabited by bacteria anyway. People used to think that it was bacterial, but now we know it's not. It's a debunked myth. The proteins they release have various effects on the blood. I believe the main one is an anticoagulant. In case you don't know what that means, it's a protein that prevents your blood from clotting, essentially thinning out your blood. That's not the only toxin they release. Other effects include muscle paralysis and hypothermia. As you can probably tell, these are useful in catching prey. All the more reason to hit the gym. I'm just kidding, they probably won't attack you if you see one. Or will they?
Because of these unusual traits in Komodo dragons, scientists are eager to sequence its DNA to see what type of genes it contains and how it compares to other creatures. In a recent scientific publication, this sequencing was discussed.
As you can probably tell, they're going to have some problem with the way scientists have sequenced the Komodo dragon's genes, and the conclusion they reached with is evolution ancestry or something. This is where it gets interesting because they are referencing a scientific literature. How exciting, we get to read a paper!
When the researchers compared the newly sequenced Komodo dragon gene that were common among reptiles, they found many unexpected traits specific to the Komodo dragon. And many of these genetic novelties were associated with its remarkable mammal-like ability to exhibit high levels of sustained physical activity.
Okay, well that one piece of text you're showing on screen there doesn't support your point whatsoever and can even go against what you're saying. The Komodo dragon genome has a similar distribution of single copy, duplicated, fragmented, and missing universal vertebrate genes as other reptile genomes. I won't go into detail on what single copy, duplicated, fragmented, or missing genes mean here. Just know that they are just different types of genes that can be classified as such. So basically, this is saying that when you compare the Komodo dragon's genome with other reptile genomes, they are similar in their percentage of those types of genes. In other words, this doesn't support your claim at all that scientists found unexpected traits specific to the Komodo dragon. If anything, this one passage here is saying that its genome is quite similar to other reptiles, which is kind of the opposite. Of course, that doesn't mean the authors didn't say that the Komodo dragon has unique genes, because they did.
Because the gene variations are unique to the Komodo dragon and very different from other reptiles, the genes were deemed to be the result of positive selection, which is a magic evolutionary phrase.
It's not a magic phrase, it's a very real thing. This takes me back to my high school days. There are 3 types of selection that describes change in gene pools. You have positive selection, which is when a phenotype moves in one direction. For example, giraffes get longer necks, or humans getting increased brain size. Then you have stabilizing selection, which is essentially where 2 extremes are unfavored by the environment. A hypothetical example would be, let's say, size. If you're too large, it's very easy for predators to spot you, but if you're too small, you don't travel fast enough to get away from predators. The last one is disruptive selection, where the 2 extreme ends are actually favored. The most popular example here is color. If the environment has 2 different colors, say black and white, that allows an organism to blend into, then it will favor those 2 colors while selecting against in-betweens like gray. Blending in is advantageous for many species in order to prevent being seen by predators. So these are 3 types of selection in which one of them is positive selection. It's not some magic evolutionary phrase.
A creature's environment has no godlike ability to create new useful genetic information for complex and multiple traits like those associated with complex metabolic functions.
The environment doesn't create it, but it pressures change. And when the organism has traits favored by the environment, say high metabolic functions for hunting, then it will be passed on to future generations until it eventually becomes the norm. I find it funny how you're against this article, but at the same time you're going to use it in an attempt to prove creation. Come on man, choose one. Either you're with this article or you're against it. Don't cherry pick whatever you want.
Evolutionists basically substitute the magic words positive selection or natural selection for something only an omnipotent God can do.
Everyone has a right to their own opinion, even if they're wrong.
It's also noteworthy that the researchers reported actually throwing out data in their selection analysis where the variation was deemed unreasonably high. The data was actually manipulated to show less variability and therefore more in line with the evolutionary model.
Ah yes, now this is where we talk a little bit about the nerdy stuff, and we'll start by discussing how evolutionary patterns are determined. Now, when people think of the evolutionary tree, they tend to think that our main form of evidence comes from fossils. And while fossils is indeed a big part of it, there's an even bigger field out there, and that is the study of DNA. There's a huge branch of bioinformatics that deals with being able to map out organisms in their respective phylogenetic trees through the comparison of DNA. This article here applies those methods in order to study how the Komodo dragon obtained certain mammalian traits as a reptile. I was interested, so I went to the article and read how they did it. Their first step was to obtain the genome of the Komodo dragon through various processes, one of which being de novo sequencing. How exactly do you get the entire sequence of an organism's genome from scratch, I hear you ask? Do we just zoom in and look at the nucleotides individually and record them down? As tedious as that would be, that's not how it works. This is where de novo sequencing comes in. In simple terms, you blast the genome into small pieces and then attempt to put them together, kind of like a jigsaw puzzle. But how do you determine where each piece fits into what position? That's where the power of computers comes in, and is a branch of the field I'm studying right now. Algorithms can be written that pieces together DNA using the overlaps that are given to them. I had to write certain code that dealt with putting together sequences of DNA, and it certainly isn't easy. Nowadays, there are tools that help out with this kind of stuff. Since the process of assembly starts with no prior knowledge of the organism's genome, we can only get it near to, but not perfectly 100% confidence, and these are based off certain assumptions. But nonetheless, it is accurate to a very high degree. Of course, that would depend on the researcher's methods, and a combination of procedures are usually done. Relying on de novo sequencing itself may not be the best idea. Anyway, the authors of this paper claim that they ended up with 1,400 scaffolds for the entire genome, which honestly isn't bad at all. There are some other information on the assembly, such as comparisons to closely related species, K-mer counting estimations, GC content percentage, and it seems they've identified individual genes too. That itself is another huge field. Being able to determine what is or isn't a gene, where it starts and ends, and the location of its promoters and exons. In other words, this is not a small project whatsoever. Anyway, after the initial step is done, along with gene analysis, they compared the genome and proteins with a select few other species that would give us information on where the Komodo dragon lies in the phylogenetic tree. There are numerous tools out there that allow you to do this, and as long as you have the ortholog protein sequences at hand, it can be very easy to do. Give them to me and I could get estimated phylogenetic trees in just a few minutes. There are tons of different softwares that do this, and they differ in the way they do multiple alignment. Knowing which tools to use at what situation is part of being a bioinformatician. Now, I was originally planning to show you guys how this is done since it's very simple, but I don't know what proteins they used, so we'll just leave it at that. It seems that the Komodo dragon is indeed a member of the reptiles, which means its evolutionary changes that give them traits similar to mammals are an example of convergent evolution, and for that to happen, positive selection must occur. This is the main part of the paper that we're interested in. 201 genes were identified to be positively selected for, most of which are orthologs with human genes. There are many ways to test for positive selection. One way is to compare allele frequency within the entire population, but since this paper only mentioned that they obtained tissues from a few Komodo dragons, that is not the case. Which leads us to the other method, comparing non-synonymous to synonymous substitutions, which is what the paper actually talked about. Synonymous meaning a substitution in the exon of a gene that does not affect the amino acid product, while non-synonymous does. These are determined by aligning the genome to that of a closely related species, something that is also incredibly easy to do. The basic concept is that if there is a high ratio of non-synonymous to synonymous mutations, especially in genes that give some sort of beneficial phenotype or reproductive advantage, then positive selection has occurred. Those are very easy to identify. And since we are comparing between multiple different species, we can assume that the individuals represent their own entire species. So let's look back at the claim you made about the researchers throwing out data to suit their evolutionary position. They threw out genes that had high non-synonymous to synonymous mutation ratios for a specific reason, and that was to get rid of false positives caused by low-quality gene annotation. This practice is commonly done in all fields of science really. You have outliers, you throw them out, because sometimes it's important to predict what went wrong, and false positives are much more common than you might think. This is not a dishonest attempt to fit the data within the pre-made conclusion. Anyway, there's a bit of the video left, so let's watch until the end.
The reality is that these genes, specific to the Komodo dragon, were engineered to produce their unique God-given traits.
You see this shit? There's so much that goes into the analysis and computational methods of determining ancestry, positive selection, and evolutionary patterns, but creationists want to dismiss all of that with nothing but the idea of God. Imagine us spending centuries developing biocomputational methods and then creationists just come in and say, no. You don't have to imagine it, because that's literally how it is. Good thing that creationists don't work in this field of science.
No sign of evolution existed in the data, even though the researchers cherry-picked it to favor the evolutionary model.
Hello, did you not actually read the paper? You know, the part about synonymous and non-synonymous substitutions? There's a whole section in the paper that talks about positive selection. Whatever, I'm just wasting my time. Thank you guys for watching, and thank you once again to Telltale for having me on. See you later.